Good morning, good evening. Thanks for listening. This is Medical Show. I respect the law and I hope you do too. The information today is for scientific and educational purposes. The slides or papers shown or discussed today will be put in the description below. And it's an open topic as usual for a while. So if you're watching this back, you might want to fast forward until you see some slides, a screen change, and then that's when approximately when I've gotten up to the topic, because at the moment there's no topic. So I'll just wait for some viewers to come in and they put in some questions or some discussion topics related to plant science, soil science and microbiology, and I can start showing slides and elaborate on the subject. So I've got quite a few slides to show and share, so I just got to wait for the topic to come up. And so I'll wait till 2 minutes for people to arrive, like Sunshine RB says hello Sunshine RB. And then after 2 minutes, which is in 30 seconds, I got to think of something to say. If there hasn't been any questions or discussion topics put into chat. And if it's another short show, which I don't mind, because it's all about the viewers, possibly next week I could elaborate on microbes, counting of them, and how to know when to use more. Yeah, it's probably an all right topic. Something along those lines, because some of my studies has been really elaborating on that, and it's fascinating. So I'm sure you'll find it fascinating too. All right, we're after 2 minutes. I'm going to talk about something or other, or maybe Sunshine's got something that's been discussed lately and wants to see if it's a myth or not. That would be fun to elaborate on that. And for those who like cricket starts tomorrow. Woohoo! In Australia, that is. Women's 2020. It's always good fun. So I'm looking forward to that. But that's not what the show's about. We need plant science, soil science, and microbiology stuff. That's what I'd like to discuss. At the moment, I don't have anything to discuss. Sunshine Rainbow says, opinion on banana peel tea for flour. Oh, so he's thinking of the potassium in it, and that might be able to be unlocked by microbes that he hopefully has in his substrate that are going to make it available to the plant. Oh, there's Scarecrow. Hello, Scaregrow 420. Nice to see you, mate. Sunshine says, apparently it hardens flour as well as makes them bigger. It hardens them. Oh, wow, interesting. I'd use gibberellic acid for that, but makes them bigger. Okay, well, the banana peel. So let's pull up banana peel nutrients and see what comes up. Where are we? Banana peel nutrients. They found crude proteins in the amount of 0.14% crude fat. That's not what I wanted. Nutrients. Mineral composition then. Oh, there it is. Mineral composition of banana peels, phosphorus. That doesn't say really much because I put the wrong word in. Mineral, M-P-O-S-I-T. So it says the mineral composition of a banana peel is phosphorus, iron, calcium, magnesium, sodium. Zinc, copper, potassium, and manganese were found in very low concentrations as milligrams per 100, so under 1 under 0.1%. So phosphorus, iron. So really what that tells me is you're trying to get more. Where's the potassium in it? I always thought banana peel had a lot of potassium, not phosphorus, actually. Here we go, ResearchGate. See how you can't trust these search engines? Dead set. Go to, it's always studies that are good, like this one I'm about to go into, or Wikipedia. It's the only things I trust. Didn't even bring it up then. Wow. A lot of bad things. So let's have a look at this. No, I really can't zoom in. So bananas were analysed for minerals, nutrition, and anti-nutritional contents. The results of the mineral content indicate concentrations, milligrams per gram of potassium, calcium, sodium. So that was actually that thing that came up in that search engine said phosphorus as the first one, remember? It went phosphorus, calcium. That's really, really bad. Gee whiz. Oh, well, lucky you got me here to decipher it for you. So at Serkins, the potassium will be the first number tested. So that's 78 milligrams per gram. So there's 1,000 milligrams per gram. So that's 0. So 1, 2, 3, 0.078. So 0.1% of the mineral content of banana peel is potassium as per this analysis. I'll put this into chat, and not in chat, in description later on. What year has this been done? Just out of curiosity. It's always good to see if it's this century or last century. 2009. That's good enough for me. Okay. So I wouldn't believe the hype. So if you want to get 0.01% potassium, there's probably better ways where you can get it, like langbeinite. I'm going to stop sharing now. Like langbeinite, that's, I got a way higher amount. What's I'm trying to say, a bit of an update. He says, I've used banana tea and results were spectacular. And I've heard the same from other people, but this isn't antidotal, not conclusive. So yeah, you've heard that it works pretty good. Well, it mightn't be the potassium in it. It might be the microbes that are actually in it. It might have some endophytes that are really rad that you're potentially culturing up. Because this seems like the mineral composition was, I was thinking potassium. So let's have a look and see what else is in there. I love these challenging questions. So you've got your calcium, which is really, it's extra low. It's mega, mega low. So the results indicate that if the peels are properly exploited and processed, they could be a high quality and cheap source of carbohydrates and minerals for livestock. So you want to see what other proteins and carbs are in there and stuff like that. Let's get down to more of it where they do more analysis. Determination of mineral content, here we go. That's how they do it. Determination of hydrogen cyanide, okay, wow. Determination of saponins, results and discussions. Let's see what they found out. The results of mineral content. Where's the table one? That'd be good to see. Yeah, here we go. Rad. Oh, that's pretty much what it did say in the small amounts was that. Now, oh, here's the next bit. This is the bit I was really after. Not moisture. Organic matter. 91% organic matter. See, just that alone is good food for your microbes. So if you're putting any microbes in the tea or if you're culturing up, just that alone is pretty cool. 1% protein, lipids, so fats, it's got 2%, that doesn't. 60% carbohydrates. So that can be utilized by microbes. And crude fiber, 30% can be broken down. That's organic matter, a form of organic matter. hydrogen cyanide, oh gosh. So why would banana tea work so well? What else are you putting into the banana tea? It can't just be banana peels only with a bubbler. You've got to activate it, I reckon. But the organic matter in itself is high moisture. That doesn't mean much for what you're trying. You're putting it in a tea, so you're seeing why is it work so well in a tea. All right, that's pretty cool. Stop sharing. I was hoping you'd update us a bit, Sunshine. What else are you putting into the tea, mate? Because it seems that apparently Sunshine's heard that it hardens flowers as well as makes them bigger. So that's pretty interesting. So yeah, I use gibberellic acid, does the same thing. Oh, here we go. Bit of an update. Sunshine says basic tea is peel in water for 9 days using airstone, and that's it. You don't put any sugar sauce or nothing like that. Just the tea and bubble it for 9 days, and then you've got a result. Wow. Well, go back to the. So it's just, yeah, just to confirm, it's only those things you're putting in, nothing else. And what sort of water are you putting in? Is it well water, or is it like mineralized water? Because if it's town water, it has minerals in it, which might also be adding to the microbes in your feed. And where'd you source the banana peels from? Another question. Are they organically grown, or are they, for instance, from the shop? Because if they're organically grown, they might have more microbes in them, where if they're from the shop, they might be killed and not worrying about the microbes. It has its own carb source. It sure does. It was said it was 60% carbs. Oh, I'm still sharing. Dechlorinated tap water, and it's organic sourced. Okay. I was going to share because I was going to get back to this page. So I can't really remember. It was organic grown, so it does have microbes inside of the bananas. And obviously, I think the water source, dechlorinated, yeah, which helps with retain microbes, and it's from the tap, meaning it's a city source, so it would have other minerals inside of it which were beneficial. So all it would do is just bump up this element in here. And you're bubbling it, so it would get rid of all of the anaerobes that could be potentially in the mixture as well. So for 9 days, it's just a good, well, like it says here, it's a carb source. I bet you, actually, why do you have to do it for 9 days? Because if you're trying to, this is a cell composition. So if you just were to chop it up, like powder it, really dry it and powder it, you get pretty similar. Well, this is at a 7% moisture. So this isn't super dry, but it is dry. Because, 6.7% moisture is low. Under 10%, 9% things go to powder. So this is a low form. So if you get it dried up, grind it, and then it would be probably you get the same results in 2 to 3 days, I reckon, rather than 9, just for that reason that you're trying to extract the things out of the cells. So you're wanting the water to mix with the organic banana stuff, not stuff, your ground up but dried banana peels, and the water would mix with that to release it from the cells, and you'd hope to get these things out of it. So you might be able to do it quicker. That's only a suggestion. Otherwise, why does it work good? Because you're extracting the organic matter is huge, so that's very beneficial for your substrate. This is feeding microbes, because I know that you're into organics and you're into retaining and feeding microbes, which is excellent. Not into synthetics, just tip it out of a bottle. And so this is good microbe food. Look at the carbs, 60% and organic matter. So just those two alone is humongous. And then you've got the extra bonuses that come out of it. The kind of high amount of potassium, 0.01%, because it's an organic source, so it's hard to get anything that's organic that's above 1%. I mean, you can get it, of course, but in general, it's hard. Let's see if there's an update. Yeah, good point. Banana peel tea. Let's have a look to see what I've got in for tea. Seems to be a bit of a topic. I'll leave that one up there. Banana tea. I'll small share since you don't. Sharing. Teas. Microbe tea. I know I got something on teas. I'm just not sure where that section is. I don't think it would be in. I wonder if it's in the plant science. No, because it's about making things. I think it's to do with microbes. That's what teas are. Let's have a skim through here. Where's my stuff on teas? No. Microbes. Actually, there's a fair bit in that biotechnology. Environmental biotechnology had some stuff that might be relevant. My substrate mix, not crime sensing, no. Teas. Teas. Where would that be? Bubbling. Microscope, no. Plant science. Tea stuff. Seeds. It's into. So I've done some experiments on that, and I wonder if it's just in my videos. Cells. So that's what we're trying to do, reproduce cells with tissue culture. Tissue culture. Preparation stabilized time. Per attempts. No. Well, that's not really what we're interested in. You got me. I'm a bit stuck. It's not a section I can go to and go click. Tissue culture. Teas. There's a lot of things to get me distracted because there's some really interesting stuff. Which I've got to stay on topic. Tea. Come on. No, it's not that. Mineral nutrition, foliar feeds. Foliar feeds. It's not tea, no. I'll show this just in case. Foliar feeding. No, that's might have something to do with. Let's have a look. Got to show something. How plants get nutrition through leaves. Oh, that's not really it. Because we all know that it happens through the different cells that actually stay on topic. Nutrition. Okay. Don't know. Well, I'll get back, stop sharing for a bit, see if there's anything written. Okay, there has, but there hasn't. All right, I'll get to sharing. I'll find something on some teas. I wonder if it's just in general. Let's have a look at this general stuff, but I don't know where to put sections. Yeah. What's that? This might jog my memory a bit. This photo. Can you open? Gee whiz, how many times have I to click that? Oh, that's the cyanobacteria collection. Oh, no. Teas. I'll just go to study then, because they went through that in organic farming. This one I think is a bit more better. Tea. I'm just going to start from the start because I think that's all I got to these slides, isn't it? Oh no, here's all the lectures. Rad. Tea, mango tea. Oh, that's the actual plant. Castings. Casting quality. Best disease, organic crop. I'll just start from the start. I've got to show something. It's kind of wasted a fair bit of time here. It's not isn't things just coming up anymore. Very good, mate. Gee whiz. What are we looking at? Tea. No. Righto, tea stuff. Castings accelerators. Look, they're all microbes. So this is probably an example. See how the microorganisms multiplied in the lab are mixed with organic wastes or your organic teas thoroughly before putting them in the vermibed. Some important fungus species are, well, the last one's not fungus, that's bacteria, because I use that one. Trichoderma. Painsilomyces. Okay. So that's an example. By getting the right microbes into your brew, you can accelerate and unlock them. Like polymixer is nitrogen fixing and phosphorus solubilizing. So if it's got, let's get to a potassium. What's a Bacillus mucilaginosus is a potassium solubilizing bacteria. So if you've got potassium in your banana peels, which you have, and they're sitting in there, you've got Bacillus mucilaginosus in the substrate. That's going to release the enzymes so that potassium can be unlocked out of that organic matter that's immobilized. So it takes it from immobilized to mobilized through the use of bacteria. And these ones are fungi. But there's an example. That helped a bit. Talking about teas. There is a good tea bit coming up here. That was a rad study. Mix up furnace. I'll use half of them. Take home message. 100% suitable for teas. The combination application of micro-inoculants into organic wastes improve the chemical and biochemical properties of their vermicastings. So by the microbes that you've gotten out of your organic bananas, which is way better, they might have been just in themselves. You might have, for instance, polymixer in there. Or probably you've got Azotobacter coccocum, which pulls nitrogen out of the air. So that's non-symbiotic. So just by having those couple in there, it'll do that in itself for you. So that's probably a reason you've got a good source of. But oh, sorry, you said other people do it too. You have to ask them, do they use organics or not? Oh, what this? I remember got in to show some more tea stuff. Fungi, viruses, biopesticides, very good. Neem, biopesticides. Isn't it amazing? Nicotine's a biopesticide. It's an extremely fast-acting nerve toxin. But yet humans consume it. Not this human. Plant protection. Nutrient management. Here we go. This might be in it. Nutrient composition of some organic resources. Yeah, isn't it amazing how cattle dung is so low? Cake, not much green in it. Back to the topic. Teas I'm trying to look for. Growth regulators. Disease. Oh, now we're going to get into pest management. Biocontrol. Use botanical pesticides? No. Here we go. Quality of organic tea. That's tea leaf, not bubbled tea. What a shame. This is organics. Why should you be in organics? Come on. Why is it going slow? Here we go. Here we go. Here's some tea stuff, finally. Nutrient solutions in hydro-organic solutions. So filtered tea newts they use there. Filtered tea nutrients. Vermicompost has that in it, and the vermiwash that comes out of the bottom has it at that composition, all those minerals. The NPK, so it's really low, but it's very high in microbes. I think this goes on to do a study, doesn't it, on the teas? Come on, because that's the thing I was picturing. And nutrient relevance in organically and conventionally grown crops. Pesticides. I'm trying for you. Optimize, no. Should we give up? Your RSC value, so important. It's that rad thing I've referred to a lot. Growth balance differential hypothesis. Big control. Righto. Looks like it's not very good. Sunshine says, I reckon it would have bloom-friendly growth regulators. I reckon it would have bloom-friendly growth regulators. You reckon it's got growth regulators in there, like what? That's pretty interesting. Hey, there's Mace. Here you go, Mace. Nice one. Good to see you, mate. So we're just talking about teas, nutrients. I was asked for my opinion on banana peel tea, and we were just going through the different compositions of it and seeing why it was good and why it works for a lot of people. Apparently, it works for lots of people. And yeah, who knows why? And if what Sunshine's going to ask the other people if they get organically sourced bananas, because it could be a microbe thing, like Gluconocidobacter diazotrophilis is one particular microbe that is very beneficial in monocrops, monoploids, diploids. Yeah, so it works really well with like sugarcane and garlic and well, with sugarcane, it supplies 80% of the nitrogen. But in normal other diploid plants, it's there's been no real test, but it's been suggested that it works really well with one particular type. Actually, I think banana is also a monocot. Yeah, which therefore that Gluconocidobacter diazotrophilis would work well with. All right. I don't know what else I can add. Let's have a bit of a look through and see if one of my other classes has got it. I'll just go through that organic farming one, because that was the organic farming certification. What am I looking at? Oh, look how many are open. What is it? So we'll go back to organic farming. Pesticide, seeds planting, crop rotation, waste management. Oh, geez, there's not many. Let's have a look. This was a very basic one of the. This was the pre-course to the other course. If it opens, there's no word to see open. You just edit now. Open. Enrichment culture. That's pretty interesting, though. This is composting. Different methods. It's the way they harvest their vermicastings. Isn't that cool? Oh, that's the route how to make it. The 3 types of earthworms. And NECioic, you don't want them because they eat. I'm going to stay on topic. Epijoic and endojoic. Epi means on top, endo means inside. That's the geosmin. Yeah, worms secrete ammonia from their pores, just like our kidneys do. Fascinating stuff. Very beneficial. This non-symbiotic Azotobacter crococcum. That's actually the species I use, crococcum. And see, it's got this. Anyway, back to the topic. Azospirillum. Oh, Lipoforium. Phosphate solubilizing microbes. That's an example of a group of heterotropic microbes solubilize the fixed phosphorus by producing organic acids, phosphatase, which is an enzyme, and makes them available to the crops. So with your phosphate that you've got in your banana, you've got mucilaginosis as an example, Bacillus mucilaginosis, that unlocks phosphates. It's a phosphate solubilizing microbe. So that's something that you would put in with your mix, and you would get that benefit. That's how it works, just by microbes. Or you can. This is the immobilized way of feeding, or you can do the synthetic way of feeding, which is tip it out of a bottle. But this is the organic approach, which I support massively. And you know why everyone else doesn't really support organics is because there's no money to be made off it. So it's really hard for people to build businesses. But it's all about sustaining the earth, being healthy. Traps, tree banding. That's pretty cool. Paste. Okay, any tea stuff? Onion. Chili. It was a good question. What do people do with their onion and chili plants? Do they put them in the compost heap? Because it kills off bugs, or it kills off microbes. Ooh, what do they do? No one's really asked that question, no. I'm trying to find some tea stuff. Just talking about the monocots versus the dicots. How the bananas are monocot, and garlic and sugarcane, and how they Gluconoacetobacter diazotrophus works well with it, which is a nitrogen-fixing bacteria, a symbiotic one. Seed planting, germination, no. No. What's that rate experiment? Absorbs oxygen. What's this experiment on oxygen? I like experiments because they prove a lot. Oxygen present. Oh, I'm not going to. The spores germinate. I don't know what it is. I'll try and stay on topic. Treatment breaking seeds, no. Bio-fertilizer, yes. Azospirillum, Azotobacter. Phosphobacteria are common. They're common in soils too. That's why a lot of these will already exist in your soil. And if you use pesticides, herbicides, etc., hydrogen peroxide, vinegar, all this sort of things that just kills all the microbes off, they might not be there. But if you've got them, if you don't do that, if you try and maintain them, the spores can last thousands of years. That's thousands of years. So if Azotobacter is sitting there and they've got fantastic resistance to spores, so it can last for thousands of years if it's not killed off somehow. So let's cut a long story short. So there's many different types that do many different things. But in the soil, as you know, there's just millions of different microbes. So the organic approach, that's why organics works, because they slowly build up all the numbers of the healthy ones. Then all of a sudden, all these are working for the farmer. It's fantastic. That looks like stop sharing. I'll see if there's any words written. G'day, Lene. Nice to see ya. Here's Shipwreck. G'day, Shipwreck. How's it going? Howdy, howdy. We're just talking about teas, and I don't have, actually I've got past shows on teas. I'm sure about it. Let's have a look in there. These are all my past shows. I'll share it. So today it was really just talking about teas and microbes. No, not microbes, sorry, we're not getting on that. But they're involved in teas, so just tea stuff. What am I doing here? Lives. I'm sure one of these had teas in it. Microbes rule, don't spray. Gee whiz. There's a few to go through. Let's see. Health, immune response, extraction. No. Righto, there was one up here that was close to it. Microbes inside and outside the plant. That was fascinating. Plants emit sounds. That's so cool. Anyway, on teas, that's what I'm looking for. Nanofolia. Smart fertilizer. Am I sharing? Yeah. Anything in here? No. Nanofolia sprays. Let's see what this. See what nanofolia sprays. That's right. I clicked on it, didn't open. So you got to do this now. You can't double click anymore. Micronutrient formats. Okay. Conventional micronutrient products sometimes lead to loss of money and unnecessary buildup of metal in the soils. Plant leaf extract can be prepared in solvents. Okay. Zinc oxide nanoparticles have been tested in the lab as bactericides and fungicides. So see that? They kill things off. They kill off, well, Bacillus subtilis you've all heard of, and Pseudomonas genus as well. So you have to be careful what your inputs are. So I would not use that. Benefits of foliar fertilization. It's not really what we're talking about. Plant growth promoters as biostimulants. Well, it's kind of it. You've got your natural ones: seaweed extract, organic acids, growth hormones, enzymes, amino acids, humic and fulvic acids. So growth hormones, you get that from your beneficial microbes. Seaweed, organic acids. You'd have fish emulsion as well in there for biostimulant. Humic and fulvic acid, yep, and humin. Smart micronutrients. No, a bit off the topic. What's your final verdict? Sunshine says, what's your final verdict on banana peel tea? Huh? Well, I reckon it's pretty good. If you get results, what sort of results do you get? Because it's a micronutrients, but it's a tea, so you're culturing up something good. So your amino acids, there's heaps of carbohydrates in it. The organic matter that's in there, which is the humic and the fulvic acids you'd be releasing from that. There'd be natural hormones, like the phytohormones, but there'd be natural ones from microbes that they've produced in there as well. So it's good. If it's good, mate. You're seeing good results. I think stick to it. But you couldn't be using it as a full time. You'd have to be putting in, like, use that as one of your tools as your trade. For instance, if you've got a 3-month cycle, you might think, all right, once a month, I'm going to do a banana tea peel blend. And then your other 3 lots a month or 2 a month or whatever you do each fortnight cycle, you might want to do something, another mix up. So you've got your different mixes of different things going in. And remember that microbes aren't always work together. Some are in competition, some are predators, some eat them. So they're not always symbiotic or co-mutual or mutual. So by doing separate teas, you're allowing that to happen. And then you're allowing your substrate and the rhizosphere for your plant's roots to choose what it wants to give signals to multiply them and have them working for your plants. So I think it's good. I'm happy that you found something rad. Just keep it done. I don't think it will work as an only thing, as an only input. It's going to be a tool in your toolbox. But you can prove me wrong. Science is awesome by doing experiments. You can just leave one plant or 2 plants where you just give it this and see what the difference is with your other strawberries or whatever you've got growing. Yeah, that's good. So yeah, I hope that helps, mate. And I'll put in chat the chemical composition of that banana peel extraction from the Environmental, Agricultural and Food Chemistry magazine. So back to Oh, he's got something. Shipwreck says, oh, nice. Tea are a solid subject. I have some I need to try out on these days, says Shipwreck. Sunshine says, try worm castings, beet juice, oat flour as carbs, and aloe vera. Awesome. And then Sunshine says, also find a nice source of leaf mold and humic matter. Oh, leaf mold? Well, what is it? The white is penicillin genus, isn't it? So you can get harmful ones in that. You'd have to leaf mold. I'd be careful introducing that into your substrate. I'd be putting that through a compost first if it was me, because you don't know if your leaf mold is pathogenic. Who knows if it's got problems in it. For the sake of doing all the right things with your microbes, you don't want to culture up a pathogen. So be careful on that one. Shipwreck says, I was always big on kelp. Yeah, kelp's rad. Green and brown kelp. That's what's got all the hormones in it. Remember, cytokinins is in the kelp, and that's one of the beneficial phytohormones that plants need. Sunshine says kelp's good for growth hormone boost. Yes, it sure is. They found that out first doing tissue culture back in the 50s. They were using some kelp, and it was working really good. And that was one of the reasons I later on found out, because of the different cytokinins that it has in it, the hormones, it's hormone boost. It's very cool. All right, tea stuff. What other stuff would you put in for a tea? You got worm castings, beet juice. Yep. Well, by having different teas going, you can put in your organic inputs, like your kelp, your langbonite, for instance, humic acid. Those types of things can go into your teas, and you can get the microbes working with them, because some of the microbes might be able to unlock those elements that are bound up because you're using organics. So it won't be a readily available mineral. It'll be bound up into something, and you need something to unlock that, or enzymes or something to allow the process to happen, like nitrogenase is good for unlocking nitrogen, or phosphatase is good for phosphorus. So mixing them up also allows microbes to have a big diverse range. So if you keep doing the same tea over and over, you can only possibly get the same amount. And if you even add microbes, some of them might die off because they're being eaten or in competition with other ones that are more stronger or powerful. Also, it's in genetics. Microbes have about 8,000 genes. So if some of those genes are also mutant genes, they mightn't work well. Getting a bit heavily into that now. Back to the tea inputs. So by having different teas, you get a different range of microbes, which is inevitably going to help in the long run. Yeah, that's the point I wanted to make anyway. So yeah, by mixing it up and you're having different organic sources, because if you for instance source oat flour from a non-organic source, that could have, or highly possible that it's got on it pesticides or maybe fine metals or production aftermath that happens from processing it, because they don't really care. But when you get organic source, they really do care, and they won't put it through these mills and other types of things, that die grinders, other things that have metal that's come off them. They've got to change their parts and sharpen their parts up. Where's those materials go? It goes into the samples. So it's just another source. Oh yeah, all inputs need to be organic. Yeah, I'm just trying to stress that point, mate. That's all. Just telling the difference if it's not organic, the difference that you get. Because for instance, copper is an antimicrobial. So if you've got copper in your tea, comes out of a can or comes out of something or other, and you get it in your mix, bang, it's all the microbes that touch that, it kills them. So the organic sources for teas are nearly a must. And if you have an option to get it from someone, you really have to ask them a lot of questions. Like if you can get it from a farmer, you have to ask them how many times do they give that animal injections, or the health of the animals. An example of that is the frogs can't remember where it was, in a stream, they were all female and staying female because of the birth pill. So the females would take the birth control pill in that area, were peeing it through, and it was going through the cycle and then into the rivers, and that was the adverse effects. So it's really, really important what you put in to your inputs to make sure that you're getting the result that you want for your outputs. Yeah, it's nearly anal. It's really, really hard, but if you can get into a good practice and get a good source, it'll be straightforward. Organics will look after you, but to get there, it's very difficult, as everyone knows. People who have tried it and people who are trying to transition, even that, they struggle because it's a lot more labor intensive, as you can see here. You got to ask more questions, try and get different sources. You don't just go to the shop and just buy it. You got to try and source it from somewhere that's done all the good right practices. And how do you know that they've done the right practices? What, because they told you? That's not good enough these days. People's word means nothing. They're all about keeping the blooming person happy. It's a blooming nuisance. I'd much rather hear the shocking truth if that's what it is, rather than yeah. So your organic source is everything. That's probably why too organic produce and everything is a lot more dearer, because it's harder to do, or harder to set up so it can cycle better. Then you just need one. Yeah, I'm not going to go into that. So Lene says organics in every aspect of life. Yes, it is if you can afford it. Otherwise, try and grow it, and even that's hard. If you go in Australia, I'm starting Bio-fertilizer to get the Bio-fertilizers, different microbes. And I asked at one agricultural shop, why isn't organics very pronounced here? And the response was, oh, organics is only new in Australia. This is at a major agricultural produce centre. Organics is only new in Australia. And I was just thinking, mate, it's, anyway, that's what they reckon. That's from, so I was just amazed. Because all my studies are, they're in India, or the majority of them are, and they've been doing these ways for decades, because they've got such a high population that they have to do these techniques to sustain the earth. If you keep doing Australian techniques, spraying everything, glyphosate, and then go and put nitrogen on and urea it up, and then that gets washed out into the streams. Oh no, it's all gone again. I'll just put some new urea. I'll just put some more on. Bang, and then it goes green again. They harvest. They think it's all good. That's the old school bad practices. I'm not going to really go into, but that's the difference. Australia hasn't adopted the new ways yet, and they're very slow, and it's just a shame. It'll come. Overseas countries are very much into the organics side of it, and I'm not saying Australia's not into it, but it's well, I've been told it's only a new thing. Yeah. Good luck. Wow, 54 minutes we've been going for already. Wow, thank you for listening. If you're still listening, these microbe stuff, they really get me. It's the future microbes. So once I start and get anything around that topic, I find it fascinating. And learning more and more about them, they work for us more and more. It's the education in Australia isn't being taught towards organics either. So the changes and practices of farmers isn't going to happen soon. It's the people that are trying to source it out that are going to get the results, and that have the organic beef and organic produce. That's who are reaping the results at the moment. But 99% of the rest of them. So hopefully they'll switch soon, transition. It takes 3 years to actually transition your farm, but you can do it in sections. That's where a lot of farmers don't know. You don't have to do the whole thing at once. Divide it into a quarter. Do it over 4 years. It's pretty cool. Once you can get into it. 7 actually organic certification bodies in Australia. Mace says, oh, that sounds good, Mace. Holy mackerel. Everyone loves mango. Huh. Far out. Wowie. It's good to see the medical system in Australia might actually be not working. Helping. Helping's a better word for it. So good on them. Good to see. Fantastic. And with another thing too, while we're on to teas, is the bubbles. Oh, I've got a lot on that. We're at the end of the show. Yeah, I'll just quickly stress this extremely amazing point. It's about tissue culture. Where are you? About the bubblers. A lot of research has been done in the fair income. I'm going to go through all in the bubbles that you put through with your microbes. I'll try to go press 2 buttons now. Why is it taking so long when I'm trying to open it? I'll open it again. Gee, the flow's not with me today. Look at that. Why? Right. Now open. Open. Second time lucky. All right, now we're getting into the bubblers. There's a lot of research that's been done on the size of it, because if you have the holes, the bubbles that are too big, they can kill off your microbes. So I'm going to get in here to the cool section, because there's been heaps. This was really stressed the point about the different aeration practices that you use. It was at the end. Maybe I should have started at the end then. How much? It's not searching fast. Oh. Here we go. Aeration and culture missions. Here we go. Right at the end. Well, that wasn't what I wanted, but I'll read that. Influence and culture conditions. Oh, okay. Aeration and culture mixing. Culture agitation and aeration is independent. They put a lot of, to the take-home message, I was going to try and prove it to you with slides, but it's the bigger the bubble and the faster it comes up, the more damage you can do. So try and, like in here, they slow it down. See the RPM, 90 revolutions per minute. So that's nearly 1 around a second. So it's 1, 2, 3. So that's how fast that the fan blades spin in there. So it's not real fast at all. And that's for the reason is for that, because it does the reduction of growth, secondary metabolite production, the shaking speed can be due to the oxygen deficiency, accumulation of CO2, or volatiles like ethylene, or nutrient transfer limitations due to the boundary layer formation. Let's see, we should have in here, if this is the right course, it's to do with the different, I don't know that blinking bit. in between it. Why does it do that now? Plant resistant. All right, I'm going to stop this. It's ridiculous. It was in, I think, environmental biotechnology then. Environmental biotechnology. Actually, do you really want to see it? All of the... It's only proven what I've just been saying. Great. It's nearly ending the show. This is a bit more technical, this one, but they might go into the aeration practices. I'm not far. No, okay, that's it. For some reason, things are working slow on my computer. I don't even know why. Names and percentages mean very little. I don't know what that meant, sorry. Renee says, it's not new to Oz, though. The first biodynamics farming method that originated in Austria in the 1920s began to take hold in Australia in the 1930s and 1950s. Yep, so 20 years, a lot longer behind in other countries. Yes, old backward Australia, that's what they call us, down under, eh? Sunshine Rainbows says, the only medical I can recommend is that place. That's good. Yes, I agree. Oh, not bad, not bad. Well, that's been all right today. I'm going to reschedule today teas. I didn't really go into much detail from what I consider. Maybe I can look in the banana study. I'll put that in. And that's really all I think on the topic in an hour. A little bit onto different beneficial microbes. Maybe I could put that at the end of the title. But that's about it. Just want to say thanks to everyone listening. Cricket's on tomorrow. Woohoo! Go the 2020. And I hope to see you all next week. We can have some other good discussions. Today was a good discussion. Thank you for the topic. It was really enlightening. It was great to go through to see what the benefits were on banana peel teas, see how beneficial they can be on plants and microbes. So it was cool. Because really when you're feeding the plants, you're feeding the microbes. And unless you're synthetic, unless you give mineral fertilizer, which all you do is pouring elements into your substrates, which I don't support. I support the organic approach. Get the microbes working for you. Microbes rule. Woo! So thanks again, everyone. Happy growing, happy breeding, and good health to you all. Hope to see you next week. Thank you for listening, and bye-bye.